Hi, folks. Welcome to the session all about sialidosis. My name is Dr. Celeste Seward. It is my pleasure to welcome you on behalf of the National Ataxia Foundation. I'd like to turn it over to my colleague, Daniel Peach, who will be welcoming you on behalf of his organization. Daniel?
Hi all, my name is Dan Peach, and I am the founder of Cure Sialidosis, um, uh, which is an arm of Cure Mucolipidosis. And I guess our charitable organization is really an advocacy group around educating and really advancing the research and also uh, a treatment for very rare lysosomal storage disorders, of which sialidosis is one, and one that I carry. We have patients, families, clinicians, researchers like Dr. Dowser with us today, and across the world. Um, and I'm really, really thankful to be here and share about sialidosis, how it relates to ataxia, and yeah, really just spread the word and let people understand about our community and what we're doing with sialidosis and um, how other people can join in this journey and help us along the way.
Thank you so much, Dan. It is my privilege to introduce our speaker today, Dr. Alessandra Dazzo. She is an internationally recognized with her pioneering research on lysosomal storage disorders and their molecular underpinnings. She earned her degree in biology and a doctorate in genetics from the University of Milano, Italy, and a PhD cum laude in medical cell biology and genetics from the Erasmus University in Rotterdam in the Netherlands. Dr. Dazzo joined St. Jude Children's Hospital in 1993, where she held the endowed chair in genetics and gene therapy. Her laboratory has made transformative contributions into the understanding and treating of lysosomal storage disorders, with a focus on sialidosis, galactosialidosis, and GM1 gangliosidosis. We'll see how much I butchered those there when you start talking, Dr. Dazzo. She identified the genetic cause of galactosialidosis, the deficiency of protective protein capsaicin A, and developed foundational mouse models for this type of research. Her team pioneered enzyme replacement and gene therapy strategies for lysosomal storage disorders and uncovered links between lysosomal dysfunction and neurodegeneration, cancer, and fibrosis. In recognition for her groundbreaking work, Dr. Dazzo received the 2017 Career Achievement Award from the ISMRD. She retired in January 2025 and now holds emeritus status at St. Jude. Outside of the lab, Dr. Dazzo is passionate about art, music, and craftsmanship and is a strong advocate for fitness and wellness. Dr. Dazzo, thank you so much for joining us today. We appreciate it.
Oh, thank you so much, Celeste, for this very kind introduction. And welcome to all participants. Thank you very much for being here in attendance. As Celeste anticipated, the focus of today's webinar is sialidosis. Sialidosis, as Dan mentioned already, is a prototypical disorders of lysosomal sialic acid degradation and processing, and is part of the large group of metabolic, primarily pediatric conditions that are collectively known as lysosomal storage diseases. Before I go into the discussion about the characteristic of sialidosis and the research we have done in quest for a proper and efficient therapy for this disease, I would like to start by giving you a short introduction of this very pivotal and essential organellar system, which is the lysosomal system. Lysosomes are absolutely essential throughout the life of an individual or an organism. They are very important and play a major role in development. They participate in cell growth and differentiation of cells and organs. They maintain tissue and organ homeostasis during adulthood, and they are certainly implicated in the aging process. Lysosomes are considered a truly multifunctional organellar system. First of all, the organelle is ubiquitous, is present in all cell types, with the exception of mature erythrocytes. The primary function of the lysosomal system is degradation or digestion of macromolecular substrates that need to be digested within a membrane. So the primary function of the organelle is compartmentalize degradation of macromolecular substrates. And all the other functions that the lysosomes exert can relate very much to this primary function as a major degradative system. The organelles are heterogeneous in size, shape, as well as composition. Not all of them are identical. They function exclusively at acidic pH, so obviously this take them apart from other intracellular organelles, because of course the organelle embed a number of hydrolytic enzymes that work exclusively at low pH, about 5. In addition, the position of the lysosomes within a cell can actually dictate their function. So lysosomes that are close to the nucleus might be slightly different and have a different function than the one that are close or in proximity of the plasma membrane. They also have the capacity to tether and fuse with all intracellular membranes and intracellular organelles. And I give you here some example. The lysosome fuse with autophagosomes, they fuse with endosomes and with the plasma membrane, but also have the capacity to only tether to other intracellular membranes, forming so-called membrane contact sites that are basically signaling compartments where exchange of ion, metabolites, or lipids occur. In addition to all of this, the content and function of lysosomes depend on the cell type and the metabolic state of the cells. Lysosome constituents are very diverse. They embed numerous lysosomal membrane proteins, starting with the important V-type ATPase complex that ensure the acidic pH of the organelle, and actually interact with another big multi-protein complex that is called the trivially lysosomal nutrient sensing complex. As the name says, this complex of proteins sense nutrients, in particular amino acid, and in that way the organelle respond by activating specific transcription factors that are responsible for lysosomal biogenesis and function. The membrane contains also lipid transporter, ion transporter, and ion channels, in particular calcium, and other lysosomal membrane proteins that serve the purpose, for instance, to internalize a substrate from the cytosol to the lysosomal lumen, or hook the lysosomes to motor proteins or the cytoskeletal proteins in order for the organelle to actually move within the cell. So the constituent, and of course, last but not least, the luminal side of the organelle enclose the whole array of hydrolytic enzymes delegated to, as I said, the degradation of very many compartmentalized substrates, and these include proteases, glycosidases, lipases, nucleases, phosphatases, and so on. So it is very understandable that this organelle or organellar system takes care of and control a myriad of cellular processes. And I just mentioned here a few. They include phagocytosis, endocytosis, the counterpart of endocytosis, which is exocytosis, in particular lysosomal exocytosis, the release of soluble as well as membrane-associated molecules, as well as cholesterol and lipid homeostasis, macroautophagy, chaperone-mediated autophagy, and so on. So it is very understandable that whatever insult that affect the correct functioning of the lysosome has a deleterious effect and lead to the development of disease. And of course, the primary examples of diseases that are just linked to lysosomal dysfunction are the lysosomal storage diseases. They comprise a large number of monogenic pediatric disorders that are caused by deficiency or either lysosomal enzymes or lysosomal membrane proteins. They are genetically and clinically heterogeneous disorders and often exist along a spectrum of clinical symptoms ranging from infancy to adulthood that often only closely correlate with a specific type of mutation and residual enzyme activity. The lysosomal diseases affect obviously multiple organs given the ubiquitous nature of these organelles and make up, in fact, for the largest group of neurodegenerative diseases in children. As of today, only few therapeutic modalities exist for a handful of LSDs that are individually very rare, but as a group obviously account for 1 in 4,000 live births. So clinical care often involves regulation of supplement, diet, medication, of course, and also lifestyle, as you will hear later on in my talk. So focusing specifically on the mechanism of pathogenesis, LSDs are now considered diseases of early aging because studying models, especially animal models of LSDs, investigators noticed that they share pathologic features of adult age-related diseases. The pathogenesis in lysosomal diseases is mainly linked to impaired processing or degradation of specific substrates, for specific lysosomal enzymes, causing their abnormal accumulation in lysosomes and other cell compartments, which eventually leads to disruption of basic cellular pathways and loss of cell and tissue homeostasis. Pathways, as I mentioned earlier, pathways affected in pediatric lysosomal storage diseases often parallel those that had been found to be pathogenic in adult diseases. And these pathways are the ones that I already mentioned briefly before. The major pathways affected and known to be deregulated in LSDs in many, and as well as in many adult neurodegenerative diseases include autophagy, endophagocytosis, calcium homeostasis, lysosomal exocytosis, apoptosis, inflammation, in particular neuroinflammation, and immune responses. Now we go to sialidosis. Let's focus on sialidosis. Sialidosis, as I mentioned, is a prototypical lysosomal storage disease. The mode of inheritance of the disease is autosomal recessive, meaning that affected individuals inherit 2 pathogenic variants in the NEW1 gene, one from each parent, and often the affected child is compound heterozygous for two different allelic variants. The parents are typically unaffected carriers. However, a very thorough population genetic studies have never really been pursued. The incidence in the population is low. Sialidosis is a very rare disease. The precise incidence is not really established. It is estimated to be 1 in 4 million live births worldwide, but we need to keep in mind, and that will become clear later on in my talk, that some of the patients might be really misdiagnosed, and because of that, their incidence, especially for the attenuated form of the disease, can be obviously, the incidence can be higher than what anticipated. So there are fewer than a few hundred cases that have been reported in the literature, and it has been described across different ethnic groups, but some clustering of cases occur due to founder mutations in certain populations, for instance, in Italy and Japan. Historically, the first patient reported was included in a group of metabolic diseases that were called at that time mucolipidosis. And this was in the late 60s, early 70s. And this is the reason why the disease is sometimes referred to as mucolipidosis 1. The first report of neuraminidase 1 as the primary enzyme defect in sialidosis was in the late 70s by a group in Germany of Kantz and co-workers. After this first report, several other reports followed, confirming indeed the primary defect of NEW1 in sialidosis. It took 10 years eventually to map the gene of NEW1 that is embedded literally within the major histocompatibility locus on chromosome 6. This location was eventually confirmed again a decade later by the cloning of the NEW1 cDNA. The sialidosis patients are loosely grouped in two main subtypes. Type 1 sialidosis was always referred to as macular cherry red spot myoclonus syndrome. And this basically summarized the major sign of the disease, especially at diagnosis. The onset of the symptom is late, but the age of onset can be variable. Is the milder form of the disease. Is a milder course of the disease, although differences you can see among patients with type 1 sialidosis with different mutations. So there are always modifying factors that influence disease outcome. It is often difficult to diagnose, unfortunately, because the disease shares the signs with other known conditions. And often this creates the difficult situation that the family have one patient with sialidosis, and then of course the patient is diagnosed far too late for the second child to be born. So what is seen in many of the family with sialidosis, including Danes, that more than one sib have the disease. There is a poor correlation between the type of mutation and the actual severity of the symptoms. And as I said before, that this can be due to the fact that the lifestyle, and other genetic and epigenetic factors or medications that the patient take, etc., could influence the severity of the symptoms. The genetic background, as I said, and lifestyle may obviously influence the clinical course. The patients have usually a residual enzyme activity, of course, and one of the characteristics of sialidosis is the presence of sialylated or sialic acid-bearing oligosaccharide in the urine. Type 1, on the other hand, is a quite different disease. The patients have also macular cherry spots. They have a much earlier onset of the symptoms. They have much more severe course of the disease. Again, a poor correlation between type of mutations and clinical severity. They have basically no residual neuron activity and also sialylated compounds in the urines. Going in details into the main clinical symptoms, as I said, the type 1 has an onset between adolescence and adulthood. The key symptoms are visual impairment, sometimes poor color vision or night blindness, myoclonus, ataxia, seizures, and I stress the fact that not all patients have these symptoms, but these are the main symptoms that are seen in multiple patients with type 1. They have gait disturbances and eye finding. Obviously, they have bilateral cherry spots on the retina and sometimes other abnormalities like nystagmus. Generally, they have mild or absolutely absent cognitive impairment and, of course, long survival. Type 2, on the other hand, can be subdivided in two different forms, congenital and infantile juvenile. The key symptoms are much more severe. The patient presents with coarse facial features like in the mucopolysaccharidosis, skeletal deformities, growth retardation, enlargement of the liver and spleen, muscle weakness, hearing loss, flat nasal ridge, intellectual disability, and myoclonus epilepsy, ataxia, and tremor. The congenital form may present as hydrops fetalis in newborns and can lead to stillbirth or survival for only a few months. Later onset type 2 may develop myoclonus and cherry spots in childhood or adolescence. So you can have a sense of the spectrum of clinical manifestations that is associated with the disease. The diagnosis of sialidosis rely on these major key hallmarks. So, of course, the primary diagnostic tool is the possibility to measure enzyme activity, which should be abnormally low, of course, in cells like chorionic villi prenatally or peripheral blood leukocytes or skin fibroblasts. But there are other key diagnostic hallmarks of the disease that should prompt let's say, a good physician to at least predict a metabolic disease. One is oligosacchariduria, so increased excretion of sialylated oligosaccharide in urine and other body fluid. Macular cherry spot, which is a central reddish area surrounded by retinal opacification, due to ganglion cells storing material. And this may be accompanied by progressive loss of visual acuity. The skeletal abnormalities, dysostosis multiplex is typical, or other skeletal changes can support the diagnosis, and neurological findings like myoclonus epilepsy in juveniles or young adults. Now, going into a little more about the characteristic of the Neu1 gene, as I said, was cloned in the late, let's say, 1990s, contains 6 exons and span approximately 3.5 kb of genomic DNA. It's a small gene. It is located on chromosome 6 within the MHC locus, as I mentioned earlier. The mouse NEW1 gene was mapped exactly in the same synthetic region in the mouse genome, namely the H2 region of mouse chromosome 17. And I give here some of the major characteristics of the gene. The first exon encodes the signal peptide of the protein. These boxes that are repeated 6 times in this case, or 5 times in the gene, and the corresponding protein encode for us boxes that are typical, for sialidosis, are shared by all of them. Their function is not clearly understood, but most likely they are responsible for substrate recognition. And also here is a part of the catalytic side and asparagine-linked glycosylation sites, because of course the protein is glycosylated. Having cloned the gene, the cDNA and the gene, of course, many mutations associated with different types of sialidosis have been mapped on the gene. What you can see, there is not a particular hotspot in the gene. They are scattered throughout the neuraminidase gene. Those are the ones that I basically summarized, that probably there are more than this, that are present in sialidosis type 2, and these are the one found in sialidosis type 1. They are mostly missense mutations, such as changing a single nucleotide that change one single amino acid. A few have few splice variants, and some nonsense mutations that give rise to truncated protein. NEW1 protein, now move to the characteristic of the NEW1 protein. NEW1 protein is 415 amino acid long, has extensive homology to other mammalian and bacterial sialidases or neuraminases. After cleavage of the 47 amino acid signal peptide at the very N-terminal end of the protein, and after glycosylation of the protein, neuraminidase becomes a molecule of about 47 to 48 kilodalton, mature enzyme. The important thing to keep in mind that is unique for mammalian NEW1 is the fact that the protein works in a multi-protein complex that we called the lysosomal multi-enzyme complex, or LMC. NEW1 associated with 2 other enzymes. One is the protective protein catepsin, and the other is which is a carboxypeptidase, so is a protease, whereas beta-galactosidase is another glycosidase. I'm sorry. So each one, beside NEW1, each one of these molecules is responsible for individual and different clinical conditions. Sialidosis, of course, is linked to single deficiency of NEW1, but you have an isolated deficiency of beta-gal that leads to GM1 gangliosidosis, whereas a deficiency, a primary defect of PPCA, lead to a secondary severe deficiency of NEW1 and partial deficiency of beta-gal. This particular observation points to the fact that NEW1 is absolutely or require association with PPCA in order to be stable and active. In absence of PPCA, NEW1 activity is reduced dramatically. And this obviously is something that we need to keep in mind. So NEW1, as I said, requires PPCA for catalytic activation, and this is, and also compartmentalization in lysosomes, and this is very clearly seen here. If you express the NEW1 cDNA in whatever cell type, you see that alone, if you overexpress the protein precipitates in the endoplasmic reticulum, depicted here in green, as really little crystals. But it's only when you co-express NU1 with PPCA, the protein is correctly compartmentalized in this punctated fashion, which are, of course, the lysosomes, and of course acquire the highest catalytic activity. The NU1 structure was determined nearly 30 years, or maybe more than 30 years after the identification of the primary defect in sialidosis. The 3D structure of the murine NU1 has been recently determined, revealing the characteristic six-bladed beta propeller structure that is shared by bacterial and viral neuraminases. These authors confirm PPCA-dependent catalytic activation of NU1. As you can see here, the activity of NU1 is really dependent on the amount of PPCA or CTSA, cathepsin A, present. NU1, the physiological function of the protein is depicted here. So the protein target the substrates, the target substrates of NU1 in vivo are sialic acid containing glycoproteins primarily, and occasionally glycolipids, but in order for NU1 to cleave the sialic acid from glycolipids, the enzyme need a detergent, a natural detergent. These are bound sialic acids. NU1 cleaves sialic acids from sialylated substrates and initiate thereby their degradation, creating a pool of free sialic acids. Deficiency of NU1 causes accumulation of substrates that retain their sialic acids, consequently impacting their biochemical properties and function. And that is the reason. Neuraminic acid or sialic acid, the most common form of this sugar is N-acetyl neuraminic acid, NU5AC. There are more than 50 natural derivatives of this particular sugar. They sit at the very end of glycan chains in different glycoconjugates. They are located, their terminal location is really a strategic position because the molecule is bulky and charged. So obviously they confer several important roles. They have several important roles, so they confirm important characteristics to the corresponding glycan where they are attached. They function as biological masks. They can protect from protease and glycosidase degradation. They can function as antigenic determinants. They are ligand for receptors. They are receptor for viruses, peptide hormones, and toxins. And they are very, very crucial in processes like cell adhesion and recognition, as well as cell-cell and cell extracellular matrix communication. So obviously, the sialic acid or sialic acid containing molecules exert or take care of many or are implicated in many, again, physiological or pathological processes, including response to viruses, immune modulation, neuronal function, inflammation, in particular neuroinflammation, adhesion, and tumor responses. So they are incredibly important moieties. And of course, this gives NU1 the very, very important role to modulate the levels and the status of sialylation of different target substrates. In order to study the disease in great details, The way to do it was to generate animal models. We have generated a mouse, Sialidosis mouse model, very many years ago. The mice recapitulate, I keep turning, the mice recapitulate the severe type 2 form of the disease. They show growth retardation, as you see here. They remain smaller throughout their lifespan, and they have reduced lifespan, about 5 to 6 months. They develop a systemic disease affecting most of the visceral organs, connective tissue, bone, cartilage, and muscle, and they develop severe CNS pathology, primarily affecting the limbic system, in particular the hippocampus. Keep in mind that these are total knockout, so they don't have any protein whatsoever. Whereas, and this is really the major difference with patients with sialidosis, because all of them have a residual neuraminidase protein, even the severe cases. So they are not messenger RNA negative. They have messenger RNA for the protein. The pathology is characteristic of characteristic of it's a bit scrambled, I apologize for that, but it's characteristic of a lysosomal storage disease. I show you the liver, the spleen, the kidney, and the bone. They all show vacuolated and ballooned cells, which are obviously the hallmark of lysosomal storage diseases, and the same also true for part of the brain, the ganglion, the hippocampus, and the choroid plexus. Now, to study mechanism of pathogenesis, my lab had focused on signs of pathogenic signs that were very well described in patients. And so we started to look at specific characteristics, for instance, enlargement of the spleen, the reason for enlargement of the visceral organs, neurological symptoms, the presence of oligosacchariduria, and the presence of myoclonus ataxia linked to potential muscle dysfunction. So, of course, the whole idea to look for mechanism of pathogenesis is to further understanding disease pathogenesis, to inform on possible successful therapeutic modalities, and also whatever we learn and understand better about the disease could be very beneficial for interpreting and be used as a readout of therapeutic efficacy. So let's start with splenomegaly. So how do we dissect the molecular reasons, the molecular pathways underlying this particular phenotype? Dissecting the molecular basis of splenomegaly brought us to learn that Neu1 was a negative regulator of a physiological process called lysosomal exocytosis. And this is the course of these studies. So we started out giving, at that time, we were trying to implement just very straightforward therapeutic intervention like bone marrow transplantation that would be theoretically could be beneficial for patients. So we started to transplant normal bone marrow or genetically modified bone marrow into the Neu1 knockout mice. And notice that out of the entire 25 mice that we have been transplanted, only 3 engrafted. All the others did not. So we wanted to look at the reason why this phenomenon occurred. And we noticed that this occurred in coincidence of an enlargement of the spleen in these animals. Looking at the bone marrow niche and the bone marrow microenvironment, we started to look at the characteristics of the bone marrow progenitor cells, for instance, and notice that they were much reduced in the bone marrow niche, but they were mobilized in the peripheral blood. We attributed this phenomenon to another important observation. Looking at the bone marrow stromal cells are important for retaining the bone marrow progenitor cells into the bone marrow niche. We notice that this molecule, VCAM1, which is a cell adhesion molecule, a vascular cell adhesion molecule 1, very important for retaining bone marrow progenitor cells into the bone marrow niche, was abnormally reduced as the animals aged in the new1 mice. Looking at the bone marrow extracellular fluid, we notice that many hydrolytic enzymes, including cathepsin G and neutrophil elastase, both of which are serine proteases responsible for the catalysis and the catabolism and degradation of VCAM1, were abnormally high in the bone marrow extracellular fluid of the new1 mice. This initial observation, because of this initial observation, we decided to look at other hydrolytic enzymes and noticed that they were all increased in the bone marrow extracellular fluid. This brought us to hypothesize that indeed the enzymes were excreted abnormally or released abnormally extracellularly via a process called lysosomal exocytosis, which is a calcium-dependent physiological process that normally the cells use to repair their plasma membrane. So what happens is that upon calcium, a spike in calcium, part of the lysosomes are recruited to the cytoskeletal network, to the plasma membrane, where they dock prior to fuse with the plasma membrane and release their content extracellularly. The docking process is completely dependent on an important lysosomal membrane protein that is LAMP1. It turned out that LAMP1 is a natural substrate of new1, remain sialylated, and accumulate abnormally in new1 cells. And that is the consequence, or let's say the effect of this accumulation. Many of the lysosomes are aligned, as you notice, and docked at the plasma membrane, ready to fuse with the plasma membrane and release their content extracellularly. The results of this is excessive release of active hydrolytic enzymes, as well as exosomes, which are small nanovesicles, in the extracellular space, with dire consequences for both the extracellular matrix and the membrane of the cells. So the new1 deficiency affected the bone marrow niche, leading to extramedullary hematopoiesis, so an enlargement of the spleen, following this particular model. What happens usually that in the normal situation, in the new1 normal bone marrow niche, we have usually a very low level of lysosomal exocytosis, and we have an excess of these little schematic molecules that are serpins, so are inhibitors of serine proteases. The inhibitors of serine proteases present in the osteoblastic zone of the bone marrow, they keep basically in check the level of serine proteases. When the serine proteases increase in the extracellular bone marrow extracellular fluid, of course, they overcome the role basically of the serpins. And of course, they take over the bone marrow extracellular fluid, leading to eventually the normal degradation of BCAM1, contributing to loss of bone marrow retention, of bone marrow progenitor cells retention in the bone marrow niche. I briefly go to another mechanism of pathogenesis, which is muscle weakness and fibrosis. We notice, and again, a similar situation has been attributed to the, or similar molecular pathways, namely excessive lysosomal exocytosis, been attributed to this phenomenon of muscle atrophy and fibrosis seen in these mice. If you notice, the muscle is affected, so the muscle cells, as you see here, are completely invaded by the muscle connective tissue that expand exponentially and progressively because the new1 fibroblast in the connective tissue, they are proliferative, they are migratory and invasive, they synthesize and depose a lot of collagen, they upregulate metalloproteases and their inhibitor, and express epithelial and mesenchymal markers. Not only that, but they exocytose lysosomal proteases as well as exosomes that are small vesicles full of signaling molecules. The exosomes from the new1 myofibroblast contain both migratory and fibrotic signals, like, for instance, Wnt3A, Wnt5A and B, and TGF-beta. And these particular nanovesicles are able to propagate, to exacerbate and propagate the abnormal expansion of the connective tissue that eventually invade the muscle fiber, basically fragmenting them, and eventually they degenerate. And this is obviously eventually leading to muscle atrophy due to this extensive fibrotic condition. The schematic of what I just said is here. So the fibroblast release not only extracellular lysosomal enzymes, but also exosomes containing these signaling molecules that eventually are able to propagate the pathologic signal to obviously healthy cells, expanding the fibrotic conditions that eventually lead to fibrosis and degeneration of muscle fibers. Last but not least, we go to mechanism of pathogenesis in the brain. The brain of new1 mice, and again I reiterate that these are total deficient mice, so they don't have neuraminidase. So these mice started to, especially in the hippocampal area, accumulate these little ponta that are represented of the amyloid precursor protein, or APP, which accumulate first in lysosome intracellularly and eventually deposit outside the cells in this proteinaceous deposit, as seen here, as the animal age from 1 month to 5 months, which is pretty much at the end of their lifespan. Now, APP turned out to be yet another substrate of new1. Accumulation of APP sialylated, remaining sialylated in lysosomes, leads eventually to increased processing of this molecule in lysosome and increased exocytosis of toxic A-beta peptides extracellularly. And this is seen also in both neurospheres cultured from neurons from the brain of Neu1 mice, as well as the CSF of these mice. They have a clear increase in the in the exocytose A-beta 42, which is let's say, one of the toxic signs of AD. So not only that, but Neu1 hippocampus bear homology and resemblance to an AD mouse model at the ultrastructural level. They have exactly similar characteristics. So the so-called dystrophic neurite seen in the Neu1 knockout is also seen in a classic AD model, and the same is true for neurofilament tangle here and here. So the mice could be, a neuraminidase total deficiency obviously could be a model of Alzheimer's, sporadic Alzheimer's disease. And in order to prove this, we actually tested whether the sera and the CSF of Alzheimer's cases had an increased exocytic levels of one of the marker enzyme that is indicative of excessive exocytosis of lysosomal content. And as you can see here, indeed, as the patients aged, obviously you have an increase of beta X, so lysosomal exocytosis in the sera, as well as in the CSF of this patient. Besides neurons, we also look at the characteristic of other cell types that could contribute to, obviously, neurodegeneration of neurodegenerative defects in these mice. And one population that was important for us to check, because it is directly implicated in neuroinflammatory responses, are the microglia. The microglia normally are, of course, delegated to many different processes, including synaptic pruning, phagocytosis, growth factor release. They usually have a small body, and they are very, very extensively ramified. Upon a neurological insult, they change shape and, of course, activity. And they become first neuroprotective, so they start with anti-inflammatory mediators to cope with the insult. They are responding eventually, become migratory, they modulate the ECM, and eventually they basically go into a neurotoxic state, which is which is characterized by an amoeboid shape, and of course very active pro-inflammatory status. Discuss briefly where we stand on therapy for sialidosis. I want to go back to sialidosis type 1. As I said, this patient population probably will be the one that will benefit the most from whatever therapy that we might develop and implement. For testing potential therapeutic modalities, we have generated a model of one of the type 1 mutations found in patients. These mice are completely normal. They have a normal lifespan. They are fertile. They have absence of splenomegaly and EMH, and as I said, the normal lifespan. The first thing that we tested, considering the dependency of Neu1 to PPCA for catalytic activation and stability, we tried to test whether a chaperone-mediated gene therapy could be suitable for sialidosis type 1. And for that, we use an AAV virus expressing PPCA, inject the type 1 model with this particular virus, and notice effective, that the therapy was effective in treating systemic organs. They increase neuron activity in the visceral organs, and they have improved tissue morphology. And this is shown here. This is the enzyme in all tissue affected and corrected, and this the increased enzyme activity in different organs. The one that is close to, very close, the type of approach that is very close to then has been inspired by him, is an unconventional approach using repurposed drugs that obviously could be potentially able to increase residual neuron activity. And we have tested several of them. First of all, of course, as I said, the recombinant VPCA, but also the HDAC inhibitor romidepsin, because we knew that the HDAC regulate neuron transcription, as well as amino acids or derivative of amino acids like tanganyl and betaine. One is cysteine, and one is, glycine, and one is leucine. We treat both fibroblasts from patients as well as the mice with betaine, and these are the results. We were able to actually show increased neuron activity in many of the patient cell strains, and in vivo, most importantly, the mice show increased neuron activity upon betaine treatment in peripheral blood mononuclear cells. That then is following a very, very strict diet and regimen that has helped him to cope with the major sign of the disease and cope with the low neuron activity, changing some of the clinical signs, including the cherry red spot and the sialylated oligosaccharide in the urine. And this is his regimen that he has used for many, many years. And last but not least is AAV-mediated gene therapy, which is the scope of the research, the main scope of the research at this moment. The therapeutic approach is very simple, is based on the generation of AAV vectors that express a neuron in combination with PPCA. We have tested this in our model with really quite impressive results. The therapy is effective in treating systemic organs as well as neuropathology, luckily enough. They have increased neuron activity, improved tissue morphology, and reduced inflammation. And this is an example of the protein in different organs and the way the mice appear that are really quite similar to a wild-type mouse. So the milestone in a very winding road has been long, but so the road has been long and winding, but the progress and the milestone have been consistent, and hopefully we will succeed to have at the end something that a therapeutic approach that will be beneficial for the patients. These are recent literature that are worth for the people maybe to pay attention to. These are my people of the last 10, 15 years or longer that has been with me, in particular to mention Lee Freemuth, Ida Annunziata, Hui Min Hu, and Janta van de Vlecker, that are the people that primarily took care of the work that I discussed today. And all my students and postdocs, people I collaborate within my institution and outside my institution, and of course, the patients who have been relentless in their quest for therapy, has helped me and pushed me to go into the correct direction in many, many occasions. Please check the Cure Mucolipidosis website, and we all should help them. The team in Boston of Miguel Sina Esteves and Heather Gray Edwards at the University of Massachusetts Medical School is the one that hopefully will take over my legacy and will continue with the search and implementation of therapy for sialidosis. And I thank you very much for your patience, and we are very glad that you could join us. Thank you.
Thank you so much, Dr. Dazzo. We are a little bit over time, but we have time for one question that I can see in the Q&A window. We have an attendee who asks, I have a question about myoclonus. Do you think it is epileptic, or does it have a subcortical origin? You had a slide on which you localized ataxia and myoclonus to the muscle, and that confused me. Do you think it originates in the muscles?
No. No, it's not. So it's a neurological. I am sorry if I confused you, but the reason why I pointed on the muscle is because myoclonic jerks are obviously affecting muscle movements, and that's the reason. But of course, the condition is neurologic.
So from a patient point of view, myoclonus is it's not a black and white. So you cannot say it's neurological, you cannot say it's muscular, simply because the signaling that is occurring from the central nervous system in the brain to any peripheral neuromuscular movement is not one directional. It's bidirectional. So because of the accumulation of sialic acid, what happens is the brain will send a signal, and every cell in the brain, all the way through the CNS, all the way through every muscle cell to its destination, fingers, arms, hands, whatever, it is getting fragmented and distorted both on the way there and on the way back. And so what happens is, because with the progression of sialidosis, one of the first things as a patient you experience is the inability to differentiate between strong and weak signaling. So what I mean by that is someone slapping you across the face, right, will give you a very similar sensation as a very slight breeze, or the wrinkle of a piece of clothing on your skin. So what that means is all of these signals go to your brain. So when your brain is saying point this way, point that way, pick up, you can't see, a soda, pick up your phone. All of these things are not one directional, they're bidirectional or multifaceted. So yes, it is a brain condition, but it's not a brain condition in a neurological, neatly defined box. It's more like saying it's a brain condition because the brain is now dealing with 100 signals for what would normally be one, and those 100 signals all come at the brain, kind of like autism, without any filter, all at the same volume, and your brain must determine which one of those signals is the one it listens to, responds to, and continues with. And that's why you get the jerk, because when the brain goes to reach for something, it will overextend, right? And then to correct, it will then pull back, but it will over, because the reaction is not, as I said earlier, the first thing to go is the differentiation between strong and weak signaling. So the first thing to correct in my clonus is strong and weak movement. So when your brain is overstressed or tired, my clonus becomes a jerk. And it's not a like an epileptic sinusoidal jerk, so like this, right, where it gets more and more intense, but an even flow. It's quite a violent jerk, right? And it's sporadic. So,
Wonderful. Thank you so much,
yeah,
Daniel. Thank you so much,
yeah.
Dr. Dazzo, for that feedback and for that clarification. We really appreciate it. Because we're a little bit over time, we'll ask if anyone has any other questions, please check out the Cure Sialidosis group. Or if you're not sure how to contact them, email any questions to research@ataxia.org. We'll make sure it gets their way and that you're able to find some answers. So again, Dr. Dazzo, Daniel, thank you so much for joining us today. We really appreciate your time and expertise and for teaching us a bit more about sialidosis.
Thank you
Thank you.
so much.
Thank you. Thank
And
you.
for everyone here who hung on for an extra 10 minutes, I hope you enjoyed this webinar and you learned something new today. Wherever you are in the world, good morning, good afternoon, good evening. Have a wonderful rest of your day. Take care, folks.
Thank you.